Okay, this is our review sheet for cell division and stem cells. So let's go. So it says the first questions are, where are chromosomes found? So chromosomes are found in the nucleus of a cell. That is where our DNA is contained. So what do chromosomes contain? They contain our DNA, obviously, which we know from key stage 3. And then what do our genes control? They control our characteristics. So for example, hair colour, eye colour, etc., etc. Okay, then we're going to look at mitosis. So if I get it up, let's go here. Okay, so mitosis, where does this type of cell division takes place? It takes place in the nucleus of the cell. And why do we use this type of cell division? So what do we need it for? We need it for growth and repair of cells. So when we cut our cells, this is how we then heal our scars. And then how do we draw out mitosis divisions? So if we're going to draw it, we start with the mother cell, okay, which is here. And I've just drawn 2 chromosomes, okay? As we know in the human cell, there could be 46. I've just gone with 2, so I'm not going to draw 46. And then what happens is, first of all, okay, our chromosomes, they duplicate, okay? And as they duplicate, if we started, say, with 2, we would then have 4. Or if we had 46, we would go down up to 92. After they duplicate, then the cell divides, okay? And the cell divides to produce these 2 daughter cells. And these 2 daughter cells are genetically identical to the original cell, or to the mother cell, they sometimes call it. Okay, so they are genetically identical to our mother cell. If you look at the outcomes then, as we have said, we have 2 daughter cells. They are genetically identical, okay? They will contain 46 chromosomes if we're looking at humans. In the exam, they could tell you how many. So in one question, they asked about a cat, and a cat has 40 chromosomes. It's just the total number for that specific species. Okay, and we also have diploid cells, okay, and diploid cells are just cells that contain the full number of chromosomes. If we then compare it to meiosis, okay, so meiosis again, same as mitosis, it will take place in our nucleus of our cell. Why would we use it though? We use it for the production of gametes, and if you can't remember what gametes are, they are our sex cells, okay, so our sperm and egg cell. Okay. Then we're going to draw out meiosis divisions. Okay. We essentially have the exact same as mitosis. So the first thing that happens is that our chromosomes, they are going to duplicate. Okay. So we started with 2, you can go to 4. In humans, they'll go from 46 to 92 again. Okay. Then what happens then, our cell divides, and then we get essentially mitosis. If I do red, everything up to here is the same as mitosis, okay? We have 2 daughter cells that are genetically identical, as we can see here, okay? But what happens is then those cells will divide again, okay? And as they divide again, so I'm going to do it in red. The cell divides again, okay? And then what happens then, that we end up with daughter cells, but they are genetically different, okay? They are different to the original cell, okay? Or to our mother cell, as we like to call it. So as you can see, they contain half the number of chromosomes, because we started with 2 and we've ended with 1 chromosome in each. If we're looking at a human cell, we'd start with 46 and we would end with 23 chromosomes in our cells. So the outcome then, we produce 4 daughter cells, as you can see, 1, 2, 3, 4. They're genetically different, as we have said. they would contain 23 chromosomes if they were in human, and they are haploid cells. And the haploid cells, because nice and easy to remember that they contain haploid, okay, half the number of chromosomes. So that would be 2 down to 1, 46, 23, etc. Then we've got stage 3 differences between mitosis and meiosis, okay. I went for more than 3 because depending on what you have, okay, these are the ones that I've got. So I've got in mitosis, we produce 2 daughter cells. In meiosis, we have 4 daughter cells produced at the end. The cells, the daughter cells in mitosis, are genetically identical, where they match and they are the same as the mother cell, whereas in meiosis, those cells are genetically different to our original cell. In mitosis, the cells end up with 46 chromosomes. In meiosis, they end up with 23 chromosomes. We call the cells in mitosis diploid cells, whereas in meiosis, they are haploid cells. Also, we can mention that in mitosis, they have 1 cell division. So if we go back, the cell only divides once, okay, divides once. Whereas in mitosis, we have 2. In meiosis, we have 2 cell divisions. The cell divides once here, and then it divides for a second time to produce those 4 daughter cells. Okay, and then lastly I had of why we need them, their function. So mitosis is needed for growth and repair of cells, and then meiosis is the production of gametes. Okay, what happens if mitosis is uncontrolled and it doesn't stop? It can cause cancer. Okay, it will just get a build-up of cells, and that then can cause cancer. If we go up, we can look then at stem cells. So stem cells are undifferentiated cells. So if we're not going to remember that, they are They are non-specialized cells. Okay, non-specialized cells. They're like our general animal cell if we were going to look. And then what's the purpose of a stem cell? This so they can specialise into a specific cell. So any cell that we need. And we're going to go into what we need them for now. So we have 2 types. We have adult stem cells and we have embryonic stem cells. So where do we find adult stem cells? We find it in the bone marrow of an adult. Okay. And then what can they be used to treat? They can be used to treat blood cancers and some other cancers that we can use. It can be used to treat diabetes, okay, where then we get the pancreas to try to produce insulin, okay? So therefore we can try and treat type 1 diabetes. And it also has been looked at to treat Alzheimer's disease. What are the benefits then of using stem cells? That they can cure a variety of diseases, okay? They can help cure many diseases which we wouldn't be able to do otherwise. Issues then is that we can have rejection of cells if they're coming from someone else, just like with transplants, we can get rejection of cells, and it doesn't always work, okay? We can't guarantee it's always going to cure an illness or a disease. Then where do we find embryonic stem cells? We find them in the cells of embryos. An embryo then is a developing baby. So we find them in the cells of a developing baby just after fertilization. Many people are opposed to embryonic stem cells, and the reason they are is by taking the stem cells from the embryo, we are destroying potential life. So in the exam, if they ever ask you why people are opposed to stem cells, you just need to write it is destroying potential life, okay? The key one here, okay? It destroys potential life if they ask why is anyone opposed to using stem cells. And I think that's it. So there's our review sheet done.